Hello and welcome my dear students and biochemistry learners. Welcome to this video on definition series in biochemistry part 9 carbohydrate metabolism. In this video I'll be talking about some very simple definitions associated with carbohydrate metabolism. So let's begin. First, glycolysis. The word is originated from Greek words. Glykos means sweet, lysis, splitting or dissolution. It is also called as Embden-Meyerhof pathway to honor the scientists. Glycolysis is defined as the sequence of reactions converting glucose or glycogen to pyruvate or lactate with the production of ATP. Citric acid cycle. The series of reactions in the mitochondria that bring about the catabolism of acetyl-CoA to carbon dioxide and water, liberating reducing equivalents like FADH2 or NADH, which upon oxidation through ETC, electron transport chain, generate ATP. It is called as citric acid cycle because citrate was one of the first compound known to participate. It's also called as the Krebs cycle because its reactions were formulated into a cycle by Sir Hans Adolf Krebs in the year 1937. It's also called as tricarboxylic acid cycle or TCA cycle due to the involvement of tricarboxylates like citrate and isocitrate. Amphibolic pathway. Amphi means both. TCA cycle or the Krebs cycle is an amphibolic pathway as it functions in both catabolism, that is the final common oxidative pathway of glucose, amino acid, fatty acid, and anabolism, provides intermediates for the biosynthesis of many compounds. Anaplerotic reactions, again obtained from Greek word ana, up, plerotikos, to fill. The reactions concerned to replenish or to fill up the intermediates of citric acid cycle are called anaplerotic reactions or anaplerosis. Cori cycle. The cycle involving the synthesis of glucose in liver from the skeletal muscle lactate and the reuse of glucose thus synthesized by the muscle for energy purpose is known as Cori cycle or lactic acid cycle, discovered by Carl Cori and Gerty Cori. Cahill cycle. The alanine formed in the muscles by the transamination of pyruvate is transported to the liver via blood. In the liver, alanine is converted after removal of amino group to pyruvate, which is converted to glucose through gluconeogenesis. The glucose thus formed is transported back to the muscles for its oxidation. This cycle is called as glucose-alanine cycle or the Cahill cycle. Pasteur effect. The inhibition of glycolysis by oxygen, that is aerobic condition, is known as Pasteur effect. The Pasteur effect is due to the inhibition of the enzyme phosphofructokinase. Crabtree effect. The phenomenon of inhibition of oxygen consumption by the addition of glucose to tissues having high aerobic glycolysis is known as Crabtree effect. Basically, this is opposite to that of Pasteur effect. Warburg hypothesis. Cancer cells utilize energy from glycolysis and they require less oxygen than their normal counterparts. This is called Warburg hypothesis. Metabolism of glucose to lactate even in the presence of oxygen is called the Warburg effect. Rapoport-Luebering cycle. In erythrocytes or the RBCs, the reaction of glycolysis catalyzed by phosphoglycerate kinase may be bypassed to some extent, and 1,3-bisphosphoglycerate is converted to 2,3-bisphosphoglycerate, also known as 2,3-BPG, by bisphosphoglycerate mutase enzyme. Then the 2,3-BPG is converted to 3-phosphoglycerate, which re-enters glycolysis and phosphate by 2,3-bisphosphoglycerate phosphatase enzyme. 2,3-bis-PG phosphatase enzyme. This pathway, which is a bypass of glycolysis in RBC, is called as Rapoport-Luebering cycle or bisphosphoglycerate shunt. The main aim of this pathway is to form 2,3-BPG that helps in hemoglobin function. Gluconeogenesis. The word itself is derived from three words. Gluco means glucose, new genesis, formation. The formation of glucose or glycogen from the non-carbohydrate precursors is called gluconeogenesis. The non-carbohydrate precursors include lactate, glucogenic amino acids, glycerol, and propionyl coenzyme A. GN or DN ratio. During gluconeogenesis, proteins are degraded and end product urea is excreted through urine. The ratio of glucose or dextrose D to urea nitrogen N in urine is termed as GN ratio or DN ratio. Approximately 1 gram of nitrogen from protein will form 3.65 gram of glucose. GN ratio is enhanced when catabolism is increased, example in insulin deficiency, starvation, etc. Glycogenolysis, splitting, breakdown. The degradation of glycogen to glucose 6-phosphate in the muscles and to glucose in the liver is called as glycogenolysis. Glycogenesis. Genesis is formation. The pathway for the formation of glycogen from glucose. Glycogen storage disease. Glycogen storage disease or glycogenosis is a group of genetic diseases that results from a defect in an enzyme required for either glycogenesis or glycogenolysis and is characterized by the deposition of either normal or abnormal glycogen in the specific tissues. HMP shunt. The hexose monophosphate shunt or hexose monophosphate pathway is an alternative oxidative pathway of glucose with major purpose of generating NADPH for reductive biosynthesis and pentose phosphate for nucleotide synthesis. This pathway is also known as pentose phosphate pathway or phosphogluconate pathway or the Dykens-Heurecker pathway. Uronic acid pathway or the glucuronic acid pathway. A pathway in the liver for the conversion of glucose to glucuronic acid, pentoses and ascorbic acid except in humans and other primates is referred to as uronic acid pathway. It is also an alternative oxidative pathway for glucose but does not generate ATP. Sorbitol or polyol pathway. The formation of fructose from glucose through sorbitol is called as sorbitol pathway. The glucose is reduced to sorbitol or glucitol by aldose reductase in many tissues except liver. In the liver, ovaries and sperm cells, enzyme sorbitol dehydrogenase oxidizes sorbitol to fructose. Essential pentosuria. Now we are slowly entering the disorders associated with carbohydrate metabolism. Essential pentosuria. This is a rare genetic disorder related to the deficiency of enzyme xylitol dehydrogenase. Due to this enzyme defect, L-xylulose cannot be converted to xylitol. The affected individuals excrete large amount of L-xylulose in urine. Essential pentosuria is asymptomatic and the individuals suffer from no ill effects. Fructosuria. The appearance of fructose in the urine. It is of two types. Essential fructosuria, which is a rare and benign genetic disorder caused by the deficiency of enzyme fructokinase. So the fructose cannot be converted to fructose 1-phosphate. Patients remain asymptomatic. The second is hereditary fructose intolerance. It is due to the deficiency of enzyme aldolase B. Fructose 1-phosphate accumulates and allosterically inhibits liver phosphorylase and blocks glycogenolysis, leading to hypoglycemia. This disorder is characterized by intense hypoglycemia, vomiting, hepatic failure, and jaundice. Galactosemia. In this, there is deficiency of enzyme galactose 1-phosphate uridyl transferase, leading to the accumulation of galactose 1-phosphate in the liver. This will inhibit galactokinase as well as glycogen phosphorylase, resulting in hypoglycemia. Free galactose accumulates, leading to galactosemia. It is partially excreted in urine, leading to galactosuria. In galactosemia, there is hepatomegaly, jaundice, severe mental retardation, congenital cataract, and generalized aminoaciduria. Pyruvate kinase deficiency. Pyruvate kinase is a key glycolytic enzyme which catalyzes the final step with ATP formation. Genetic deficiency of this enzyme reduces the rate of glycolysis and ATP formation. Inadequate energy affects the structural integrity of the cells, leading to excessive destruction causing hemolytic anemia. Hexokinase deficiency. A genetic defect in hexokinase of erythrocytes reduces the amount of oxygen available for the tissues, resulting in anemia. Reduced activity of hexokinase, the first glycolytic enzyme, results in low concentration of 2,3-BPG, that is 2,3-bisphosphoglycerate, that allows hemoglobin to release oxygen in the tissues. Therapeutic use of glycolysis inhibitors in cancer. Some inhibitors of hexokinase like 2-deoxyglucose, that is 2-DG, ionidamine, 3-bromopyruvate have shown promise as chemotherapeutic agents by preventing the formation of glucose 6-phosphate. These compounds not only deprive tumor cells for glycolytically produced ATP, but also prevent the formation of pentose phosphate, without which cells cannot form nucleotides essential to DNA and RNA synthesis and thus cannot grow or divide. Out of these molecules, 2-DG, that is 2-deoxyglucose, was in news recently as an antiviral component, antiviral molecule for coronavirus as well. So same 2-DG. Lactic acidosis. The accumulation of lactic acid in the blood to the levels that significantly affect the blood pH. Lactic acidosis. The high blood lactate levels can result from an increased formation or decreased utilization of lactate. Elevated lactate levels are associated with anoxia. GPD deficiency. A genetic defect, that is an X-linked recessive trait, in the deficiency of enzyme glucose 6-phosphate dehydrogenase or GPD results in drug-induced hemolytic anemia. The drugs could be like antimalarial or sulfa drugs, etc. GPD deficient persons will show increased methemoglobin in circulation even though cyanosis may not be manifested. Also, GPD deficiency offers resistance to Plasmodium infections, that is malaria. Once again friends, I am thankful to all these resources for helping me to make these videos. Thank you friends, thank you for watching this video. Be happy, healthy, stay safe, and remember, biochemistry is easy. Thank you so much.